Hello, everyone. It's my great pleasure to be sharing my work with you today. I'd like to start by thanking the program committee for giving me this opportunity. In systems and cognitive neuroscience, we investigate how sensory input gets processed by the brain under different task demands to generate motor output that indicates perceptual or cognitive behavior. At the same time, the brain generates something that is elusive but also so real. That is your conscious experience of the sensory input, also called perceptual awareness. Perceptual awareness is by no means a veridical capture of sensory reality. For example, if you are watching my talk on a large-ish monitor, you'll see that this picture appears to be moving, even though it's completely stationary. Similarly, with these classic ambiguous bistable figures, even when you fixate your eyes in the center of the image, your conscious perception of the image can't help but flip back and forth between alternative interpretations. These examples demonstrate that our classic thinking about how perception comes about, which is that input enters the brain and goes through a series of transformations to generate perceptual outcome and motor output, is at best incomplete. But instead, conscious perception can be generated by your brain's intrinsic dynamics in a way that is dissociated from sensory input. In other words, we have a revised framework now, which is that sensory input enters a brain that is always active, and it interacts with the pre-existing brain state to generate perceptual awareness. The pre-existing brain state is defined by both the ongoing brain dynamics, intrinsic brain dynamics that fluctuate from time to time, and also the connectivity patterns between neurons and neural circuits that are influenced by genes, development, and past experiences. This revised framework fits very well with what we know from anatomy. We know that in the cortex, the feed-forward pathway represented in this serial transformation of information accounts for only a minority of the synapses. The vast majority of the synapses come from horizontal and feedback connectivity. For example, in primary visual cortex, the first stage of visual processing in the cortex, less than 1% of the synapses come from the feed-forward pathway from the visual thalamus, the LGN. In the LGN, similarly, less than 20% of the synapses come from the feed-forward pathway from the retina, and the vast majority of the synapses come from the feedback from the V1. So today, I'd like to share with you our recent research that demonstrates that conscious perception results from an interaction between sensory input and the pre-existing brain state. Specifically, we have found that large-scale neurodynamics during conscious visual perception can be described as initial state-dependent robust transient dynamics. And there are two distinct spontaneous processes that influence conscious perception in doubly dissociable manners. And lastly, past experiences can leave a trace that persists through the resting brain and influences your conscious perception in dramatic manners. Before I dive into these topics, I'd like to tackle some questions that are of broader generality and interest than the conscious perception topic per se. That is, how does task processing modulate spontaneous activity? And how does spontaneous activity influence task processing? At this point, we have a wealth of knowledge from resting state studies. We know that the brain is always active. The magnitude of spontaneous activity fluctuations is huge. Spontaneous activity is highly organized in space and time. And resting state activity has great behavior and clinical significance, and disruption thereof predicts clinical deficits. At the same time, through investigations of stimulus or task-evoked activity, we have a lot of knowledge about neuronal activities involved in perception and cognition. For example, in the visual system, we have a wealth of knowledge about the neuronal response properties at every stage of the visual hierarchy and the roles of feed-forward and feedback pathways. But at the moment, there is sort of a gap between these two fields. And we don't really know how ongoing activity participates in task processing and how perception cognition arises from a brain that is always active. So I'd like to share with you some work we have done on these questions. First, how does task modulate spontaneous activity? Here I'm showing you two fMRI activity traces from the same subject, same brain area. The left motor cortex. One of them is during a task that involves pressing a button with the right hand, the contralateral hand, whenever a target appears. So it's a task that activates this brain region. And another activity trace is during resting state, when the subject is not doing any task in particular and lying perfectly still. So make a guess for yourself which one you think is from rest and which one is from task. So you can see that the peak-to-peak fluctuation in the bottom trace appears to be larger than the top one. So from the idea that motor activation should activate motor cortex and induce larger fluctuations, you might think the bottom one is from task. But it's actually the opposite. So here I've superimposed the times of the stimulus occurrence. Every red mark means the time the stimulus occurred. And you can see that after the cue, there tends to be an increase, uptick of the fMRI activity, and that corresponds to the motor activation that we know this region involves in. But overall, the fluctuation magnitude seems to be reduced. In fact, it's not just motor cortex. In this visual detection task, we find that across pretty much all of the activated and deactivated brain regions, the variance of the activity across time, in other words, how much the activity fluctuates over time, is reduced during task. What this says is that activation and deactivation both appears to be quenching the variability of spontaneous activity. We also found that the autocorrelation of the ongoing activity, how much the activity is correlated over time, also reduces during task activation or deactivation. Next, how does spontaneous activity influence task processing? Our classic framework says that during task, the ongoing activity persists uninterrupted. And every time a stimulus or task demand is imposed on the subject, that triggers the evoked activity that adds linearly on top of the ongoing activity. Although the evoked activity might have variation over time, our classic thinking is that that does not interact with the ongoing activity. And in other words, if I look at a cross-trial correlation of ongoing and evoked activity, that correlation is around 0. And that assumption is what motivates the assumption we take when we do trial averaging to extract the evoked activity, because if ongoing and evoked activity do not interact, then by averaging across trials, we can average away this random fluctuation of the ongoing activity and extract the evoked activity. There is a way to test this framework that is through a mathematical law called the law of variance sum. This law says that if I have two variables, x and y, two time series x and y, the variance of their sum, sigma squared x plus y, so the variance of the sum of the two signals is equal to the variance, so the sum of the variance plus a third term. So if the correlation between ongoing and evoked activity is 0, the third term goes away. And what you have is the well-known maxim that for two independent variables, variance sum linearly. And because variance is always a positive variable, so all of these sigma squared values are positive, that predicts that if linear superposition is correct, the variance of post-stimulus activity, which includes the sum of spontaneous and ongoing activity, should be larger than the variance of pre-stimulus activity. Is that correct? So I tested that through the data set that I just told you about. when we average across trials, we find that there is bold activation across different brain regions involved in this task. When we look at across trial variability, that is, we align all the trials to stimulus onset and simply calculate variability variance across trials at every time point. We find that across trial variability reduces for all of these brain regions. And this is a much larger effect size, about 10% of variability reduction compared to the trial averaged response. So this means that the variance of post-stimulus activity is smaller than the variance of the pre-stimulus activity. And according to the law of variance sum, that means that the correlation across trials between ongoing and evoked activity must be negative, and negative enough to offset any variability in the evoked activity itself to produce this variability quantum effect. More intuitively, what that means is that when the ongoing activity is low, you'll have a larger activation. And when the ongoing activity is high, you'll have a smaller activation caused by the same stimulus. So we found that task activation and deactivation quantize the variability of spontaneous activity fluctuations. And there's a negative interaction between pre-stimulus ongoing activity and evoked activity. This is the scenario I just told you about. There is a more dramatic scenario, which is that when the pre-stimulus activity is low, you might have an activation, whereas when the pre-stimulus activity is high, you might have a deactivation. And in fact, we found that this does seem to be the case for some of the brain regions. So that means that if you average across trials, you'll average that away. So this interaction framework still postulates that the post-stimulus activity is a sum of ongoing and evoked activity. However, that evoked activity negatively interacts with the pre-stimulus activity. But if you put yourself in the perspective of a neuron or a brain region in the brain, you realize that they have no idea of knowing when the stimulus occurred or doing trial averaging. So this partition of ongoing and evoked activity in the post-stimulus period is something that's artificial, that we investigators do. A post-synaptic neuron, what they receive from the presynaptic neuron is simply an activity trace that varies over time. And from the many presynaptic neurons that impinge on this postsynaptic neuron, its input traces a trajectory in a high-dimensional state space. So a more parsimonious way to think about the data I just showed you is that when the stimulus occurs, that trajectory of cortical activity in the state space gets constrained into a more restricted subspace. And that will automatically give you a cross-trial variability reduction, variability quenching, and this negative interaction phenomenon I just described. Because when the pre-stimulus activity is low, it might need to go high to reach this targeted subspace. And when it's high, it might need to go down to reach the same subspace. In fact, this idea of trajectory-based processing has been influential in some of the animal literature for years. For example, in the premotor cortex of macaques, during motor preparation, you find that population activity exhibit these trajectories that converge in the state space. In the rodent auditory cortex, it was found that spontaneous activity outlines a subspace in the state space within which the evoked activity visits. So these smaller circles are the evoked activity driven by different different stimuli input. It's important for me to emphasize that variability doesn't always have to go down after stimulus input. This is an example from the leech nervous system. So in this system, the same stimulus can trigger different types of behavior, swimming or crawling. And population neural activity trajectories in the state space diverge depending on which behavior is exhibited. So if you look at a cross-trial variability across all of the trials, it would go up after stimulus input. Okay, so now I'd like to take those insights I just described and tell you about what we have found about cortical trajectories during conscious visual perception. So to ask the question, what kind of neural activity differentiates conscious from unconscious processing of the same stimulus, we use the threshold level visual perception task. In this task, subjects fixate on a cross in the center of the screen, and after a random delay period, at a time that's unpredictable by the subject, a stimulus comes on. It's a ball patch pointing to either upper left or upper right. On every trial, the orientation is randomly drawn from these two possibilities. The contrast and duration of the stimulus was titrated to the individual subject's perceptual threshold, such that the same stimulus is consciously perceived in about half of the trials. After another blank period, we ask first the subject to perform a forced discrimination of the orientation. It's a ball patch pointing to upper left or upper right. And if you didn't see it, make a guess. And then we ask, did you see it or not? So these two questions probe objective performance and subjective awareness. We collect these two performance measures on every single trial. So because the stimulus was titrated to the perceptual threshold, the same identical stimulus is shown for each subject across all the trials. And that titration means that, as expected, about half of the trials, subject answered that I saw the stimulus. If you look at the objective performance, the subjects is about always correct about the orientation when they see the stimulus. Interestingly, when they answer I didn't see the stimulus, their performance drops as you would expect, but it's still significantly above chance level. So that suggests that the stimulus that is not consciously perceived nevertheless was processed by the brain to the point of influencing objective performance. So we measured large-scale neurodynamics during this task using MEG, which is a method to measure across the whole brain neural dynamics with millisecond timing. So it records the magnetic field produced by population neural activity. And if we look at the frequency content of the MEG signal and plot it in a log-log spectrum, we see that it follows roughly a power law distribution, meaning that it's a straight line in the log-log plot, and power falls off as inverse of frequency to a particular power exponent. And on top of this 1/f slope, there are peaks at different frequencies that correspond to the well-known brain oscillations, for example, theta, alpha, and beta oscillations. So across several studies, we have found that the low frequency component of the broadband activity, so-called the slow cortical potentials, appears to be a very strong correlate of conscious perception. In fact, if we just low-pass filter the broadband signal at less than 5 Hertz, we can predict whether the subject consciously perceived the stimulus or not. So to give you a little more background of this signal, it is in fact a very good correlate of resting state fMRI signals. We found that using intracranial recordings and fMRI in the same subjects, we found that across all the different brain region pairs, correlation computed using SCP and correlation computed using spontaneous fMRI signal correspond to each other very well. In other words, the resting state network that we find with fMRI is recapitulated in this SCP band. This is less than 0.5 Hertz, but if you use less than 5 Hertz, it's a very similar result. And a later study from Sheila Calholt's group also showed that we simultaneously recorded spontaneous fMRI signal and SCP signals. They correlate with each other with a time lag of about 4 seconds. If we, the first thing I'd like to show you in this task is a classic trial averaged baseline corrected event-related field responses. Here is a response from a single subject example. Different traces, different colors are different sensors. There are 275 sensors covering the whole brain. And I've sorted them by condition, by the combination of subjective awareness and objective performance behavior on every trial. So these are the seen and correct trials. There were very few seen and incorrect, so those were discarded, and unseen and correct and unseen incorrect. What you can see is that after the brief stimulus presentation, if the subject perceived the stimulus consciously, there are these widespread, long-lasting SCP activity that distribute across frontal parietal and temporal cortices. So this is a classic ERP approach where we average across trials and correct by the pre-stimulus baseline. But we wanted to see how does the activity evolution from pre-stimulus to post-stimulus period underlie conscious processing and differentiate conscious processing from unconscious processing. To that end, we took the whole head MEG activity, and at every time point, we used a multivariate decoder to predict the subjective awareness outcome of the subject, seen versus unseen. We do that at every time point from 2 seconds to 3 seconds after stimulus onset. And we found that just by low pass filtering the signal at 5 Hertz, we have beautiful prediction of single trial perceptual outcome shown in magenta here. And it's significant from actually all the way to 2 seconds before stimulus input to 3 seconds after stimulus input. The amplitude envelope of this activity also provides significant decoding, but mostly in the post-stimulus period. Using higher frequency activities, the decoding accuracy was significant in some cases, but much, much lower than the SCP band. So at every point in this case, we have a decoder that tells us, given this identical threshold level stimulus presented to the subject, we can predict whether the subject is going to consciously perceive the stimulus or not. And using the decoder, we can get a sense of where in the brain or which senses contribute most to the decoder. And we find that in the pre-stimulus period, frontal and temporal sensors appears to contribute the most. And after stimulus input, there is a feed-forward sweep of activity from occipital to temporal regions, as you would expect. A very important question here is how stable or how fast is the activity underlying conscious perception changing over time? To address that question, what we could do is to train the decoder at each time point and test it not only on the same time point, but on all the other time point during the epoch. And that's what we get here using the raw SCP activity. We find that in the pre-stimulus period, if we train the decoder at any of these time points and test it on any other pre-stimulus time point, the decoder performs equally well. That means that there is a relatively stable activity pattern in the pre-stimulus period that differentiates between seen and unseen trials. In the 1 second window after stimulus input, we find that this decoding narrows down to the diagonal. So in other words, the activity pattern differentiating seen and unseen trials don't really generalize across trials, and it's changing very fast across time. In the very late period from 1 to 3 seconds, again, we find good across time decoding generalization. And we think this is likely related to the working memory maintenance of the stimulus information in anticipation of the response. And the response comes between 3 to 6 seconds after stimulus onset. So during the conscious perception processing, it appears that the activity is changing fast. So how do we think about this in the trajectory view? It is sometimes not always easy to get intuitions from decoders. We wanted to be able to visualize the data in a more intuitive manner. So to do that, we took the 273 sensors we have and performed dimensionality reduction using PCA here. And I'm going to show you results from the top 3 PCs for visualization. But for statistical tests, we use the top 5 PCs because the top 5 PCs explain about 75% of the variance in the data. This is a very similar approach as animal work has done for looking at the state space trajectories of population neuronal activity. We found, just by doing this dimensionality reduction, we found a very dramatic pattern of results. This is from a single subject plotting the activity trajectory in seen trials shown in red and blue here, and unseen trials showing gray here. This is the same activity, it's the same result, just color-coded differently, highlighting the unseen trials. And each trajectory is plotted from 1 second before stimulus input to 3 seconds afterwards, and the circles indicate stimulus onset. So what you can see here is that the before stimulus input, seen and unseen trials already reside in different locations in the state space. And depending on this initial state, the identical stimulus input triggers the brain to travel on completely different trajectories. In the seen trials, the brain activity travels on a fast evolving trajectory that goes into distant space, distant locations in the state space. And in unseen trials, it follows a completely different trajectories. And sorry, I forgot to mention the left and right here means the left orientation or the right orientation of the ball patch. And stimulus orientation here did not have a big influence, but seen versus unseen perceptual outcome had a huge influence on the activity trajectories. This is I have a trial averaged trajectory for each of the conditions, the 4 conditions here. In fact, we can see this effect even at the single trial level. So here is single trial activity trajectories from an example subject, and black is seen and red is unseen. Each trajectory is again plotted from 1 second before stimulus input. Those are the squares here to 3 seconds after. We can see that at the single trial level, seen and unseen trials already occupy different subspaces in the state space. And that's what gives us single trial decoding that I showed you earlier. So I've shown you that in the state space, in seen trials, this activity is traveling fast to distant locations after stimulus input. A technical term for this is transient dynamics. It's changing fast, it's moving fast. There's a critical question here which is whether this transient dynamic is stable, unstable, or robust. So unstable would mean that if, for example, the system is chaotic, then small variations would blow up over time. On the other hand, a fast-changing activity, although it's evolving fast, could also be locally attracting or robust. So one way to look at that is using the across-trial variability measure I showed you earlier. So these are alternative hypotheses. In one case, if noise becomes amplified over time, variability would increase. On the other hand, if transient dynamic is robust, it would be resistant to small perturbations, and it would have variability reduction. We found that in seen trials, there was a substantial variability reduction up to 20%. And there is smaller variability reduction in unseen trials. So this suggests that these transient dynamics are robust. To summarize, what we found is that there are 2 robust trajectories in the state space. And depending on where the initial brain state is, so the pre-stimulus activity, where it is in the state space, if it's near the basing, attractor basing of one trajectory, then after stimulus input, brain activity would travel on that trajectory. And if it's near the attractor basing of a different trajectory, brain activity would travel on different trajectory. And each corresponds to a different outcome of subjective awareness, seen versus unseen. So this suggests that neural activity underlying conscious perception is dependent on initial state. It's also robust and transient. If you think about across-trial variability, if you lump all the trials together, variability would go up because the system has 2 different behaviors driven by the same stimulus input. But if you look at each individual one of the conditions, there is a robust trajectory that gives you reduced across-trial variability. So it gives an interesting phenomenon where neural activity is both robust to small variations in the initial state, but also sensitive to large variations. So I'd like to next share with you some work that we did to understand what is the source of this initial state influencing subjective awareness, and what is exactly its impact on behavior. There are different sources of spontaneous activity, as we know. On the one extreme, there are neuromodulatory influences correlated with, for example, arousal that modulates spontaneous activity across widespread cortex. On the other hand, there are also spontaneous activity fluctuations at a very fine-grained level, even at the level of orientation column. So at the moment, we don't really understand what is the specific behavioral impact of spontaneous activity at all these different levels. To show you kind of the summary of what we found in this study, we found that there are two different types of spontaneous activity that have distinct impact on behavior. So in one process that we call non-content specific, it's going to influence your subjective recognition regardless of what's the stimulus input. And it does so by shifting the criterion in perceptual decision making. And a different type of spontaneous activity is content specific. That means that one particular activity pattern promotes subjective recognition for a particular type of stimuli, such as animals, and a different activity pattern promotes recognition for different type of stimuli, such as houses. And this type of content-specific spontaneous process promotes influences subjective recognition by enhancing sensitivity. And I'll explain what this means later. The task we used here is very similar to the first task I showed, except that the stimulus now, instead of garbage, we use meaningful stimuli that occurs in the real world, such as animals, face, objects, and houses, presented in front of an ecologically valid background. And we again titrated this stimuli contrast to the individual subject's perceptual threshold, such that for each subject, each particular image is shown with identical contrast across trials. And again, subjects answer two different questions probing objective performance, category discrimination, and subjective awareness, subjective recognition on every single trial. So when these stimuli are presented because they are titrated to the perceptual threshold, the same stimulus may be seen on some trials and unseen in other trials. When it's reported seen, yes, it's a hate trial. And when the subject did not see the stimulus, we can call that a miss. We wanted to get a sense of what is the baseline tendency for a subject to answer yes. So we also included scrambled trials, which are the same images but face scrambled. So they preserve category-specific low-level image features of the original images, but they are devoid of any meaningful content. So in that case, if the subject says, yes, I saw meaningful content, then it's a false alarm. If they say no, that's a correct rejection. So hate rate in this task, which is percent recognized for the real images, is 50%. That's as intended because we titrated the stimuli to reach 50% recognition. It's interesting that the false alarm rate in this task is quite substantial. It's around 17%. And we have some data suggesting that this is not the subject pressing the wrong button, but actually indicates genuine false perception of seeing a meaningful content out of a meaningless pattern, which you can read in the paper. I won't go into the detail there. So we wanted to know what kind of spontaneous neural activity impacts this subjective recognition behavior. Our first hypothesis here is that there is a spontaneous process that influences your subjective recognition regardless of the specific stimulus category. So that would mean that we could use the pre-stimulus activity to, if that's correct, it would mean that we can use the pre-stimulus activity to predict subjective recognition status, as we did in the previous study, but also this activity pattern we find would be robust to the stimulus category. In other words, if we train the decoder in one category, it should be able to perform equally well in a different category. So this is using the whole head MEG activity we acquired in this task to predict seen versus unseen perceptual outcome in the real images. And importantly, if we train in one category and test using left out data from the same category, we have about the same performance as if we train in one category and test on a different category. So this suggests that this activity pattern is really category non-specific. In other words, not content specific at the level we are probing. So if we look at the scrambled images here, we find that we can also use pre-stimulus activity to decode subjective recognition outcome in scrambled image trials. And again, if we train the decoder in real image trials and test it on scrambled image trials and vice versa, we can accomplish significant cross-condition decoding. What this means is that there is pre-stimulus activity patterns involved in this non-content process that influence your subjective recognition of images regardless of what's the content of the image, what category it is, or if it's even scrambled. Our second hypothesis is that there is a content-specific spontaneous process which affects only your subjective recognition for specific stimuli categories. So one category activity pattern would predispose the subject to say yes if the stimuli are animals. A different activity pattern in the pre-stimulus period would predispose the subject to say yes to the a different stimulus category. So if that's the case, what that means is that so this drawn here, the blue trace indicates the spontaneous activity pattern that enhances house recognition, and green trace indicates the spontaneous activity pattern facilitating your recognition of animals. And if this hypothesis is correct, what that would mean is that if we take the SING trials, the YES trials, we should be able to use the pre-stimulus activity pattern and predict, decode which stimulus category was shown in that trial. So even though the stimulus category was randomly drawn on every trial and is unpredictable objectively, if we take condition on the SING trials, if this spontaneous activity process we postulate exists, it would mean that we can use pre-stimulus activity to predict upcoming stimulus category. Indeed, that's what we found. In SING trials, we were able to perform this discrimination, this decoding. Pre-stimulus activity predicted stimulus category in SING trials, but not in unsing trials, suggesting that there is a pre-stimulus spontaneous activity process that influences subjective recognition in a category selective category specific manner. And this summarizes what we found there. A non-content specific process fluctuate over time, and regardless of the objective category of the stimuli, if it's high, subject would be more inclined to say, yes, I recognize the image. And there is also a content-specific process that actually varies across different stimulus categories. If the face-promoting activity pattern is higher, and the stimulus is also face, the subject would be inclined to answer yes. But if the spontaneous activity is encoding a different stimulus category when the stimulus arrived, then the subject would be more likely to answer no, I did not recognize the stimulus. So in the task we have here, there is real image containing real objects and also scrambled images containing noise. So an important way to think about this type of data is that there is a neural representation, internal representation of the stimuli, and the subject has a criterion for answer sing versus unsing using the signal detection framework. If the stimuli is a real image and the internal representation is above the criterion, subject answer yes, and that's a hit, and vice versa, that would be a miss. If the stimulus is scrambled noise image and the subject answer yes, that would be a false alarm trial, and otherwise it's a correct rejection trial. There are two different ways that spontaneous processes could influence subjective recognition. One is changing criterion. For example, if the criterion is lowered, then the subject would be inclined to say yes, regardless of whether it's a real or a scrambled image, and we should have increase in both hits and false alarm. If a different way that spontaneous activity could influence subjective recognition is to change sensitivity. In this case, neural representation for the image, for the signal and the noise, would have larger distance between the two distributions. So we tested how does each spontaneous process influence subjective recognition? Does it change subject's criterion or sensitivity? To do that, we split our trials into high pre-stimulus non-content specific activity versus low. And we also split our trials based on high versus low pre-stimulus content-specific spontaneous processes. And we found that for non-content-specific spontaneous process, it influenced subjective recognition by shifting criterion into the more conservative, more liberal direction. So that means that there is going to be increased hits and increased false alarm when non-content specific activity is higher. And that's why we were able to decode subject recognition outcome using both the real and the scrambled image trials earlier. For the content-specific process, we found the opposite. We found that it increased the sensitivity of subjective recognition, but had no effect on criterion. So the fact that non-content specific activity promotes subjective recognition regardless of the content of the stimulus input says that it might be related with widespread brain activation that's modulated by arousal. There is a peripheral measure into some of these neuromodulatory influences, that's pupil size, which indicates cholinergic and norepinephrine neuromodulatory influences on the cortex. And recent animal literature has shown that moment-to-moment pupil fluctuations can indicate fast changes in cortical arousal modulated by subcortical neuromodulatory influences. So we looked at how these pre-stimulus spontaneous processes relate to the pre-stimulus pupil diameter recorded on every trial. And we found that the non-content specific process correlates substantially with pupil size in the pre-stimulus period recorded during the same time window, whereas the content specific process had no relationship with pupil linked arousal. To summarize, what we found in this study is that there are two different spontaneous processes, non-content specific and content specific. One of them, the non-content specific process, influences your subjective recognition regardless of the identity of the stimulus input. Whatever category it is that we tested, even if it's scrambled, it would influence your subjective recognition. And it does so by shifting your criterion towards the liberal direction. And it correlates with pupil-linked arousal. A separate content-specific process influences your subjective recognition of specific stimulus categories. One activity pattern promotes recognition of animals. A different activity pattern promotes recognition of houses. And this spontaneous process influences your subjective recognition by enhancing sensitivity. And it does not correlate with arousal. So this is only a beginning. There is a lot of work to be done still to figure out exactly what are these spontaneous processes, but it shows us that there are different ways that spontaneous activity influences behavior, and we need to start teasing that apart and link them to different spatial scales. So lastly, I'd like to share with you some work we've done recently about how past experiences influence conscious perception by leaving a trace in the resting brain. So we all know the enormous influences past experiences have on perception. For example, we recognize these degraded letter strings and digits effortlessly because we have a template in our brain that's matched onto this distorted or degraded stimuli. For the same reason, we find cartoons interesting and engaging because we project our internal brain states onto these reduced stimuli. And if you have seen this picture before, you likely recognize a dog in the picture effortlessly. And when that happens, once you recognize this image, it is impossible to unsee the dog in the picture. So we probe this phenomenon by creating images like this. That's called Moni images. If you haven't seen my talk before, this picture probably looks like meaningless black and white patterns on the screen. But if I show you this picture, now you might see a dog out of this picture. And if you do, that effect is likely to be very long lasting. And if you see this picture again, several months later, you probably still recognize a dog in the picture. This is not just a curious phenomenon. It has a lot of important clinical ramifications. For example, in this task that I just showed you, in fact, patients with psychosis perform better than matched controls here is discrimination sensitivity of that black and white picture before seeing the grayscale picture. And patients and controls are similar. And after learning, after showing subject the original picture, patients perform better than controls. And this performance benefit predicts perceptual schizotypy on an individual basis. This fits with the current thinking in predictive coding that when feedback overwhelms, feed-forward activity in perception, hallucinations might occur. When feedback overwhelms feed-forward in cognition, delusions might occur. So we studied this phenomenon and tried to understand what's the neural activity underlying this dramatic one-shot perceptual learning paradigm using 70 fMRI. We had each subject viewing 33 different sets of MONI images and their corresponding grayscale pictures. Images were interleaved and shuffled, so the same grayscale picture doesn't immediately follow its corresponding MONI picture. So each MONI picture is presented 6 times before and after disambiguation, and we find that percent recognized in the pre-disambiguation stage increases a bit, but not very much. And after seeing the grayscale picture, you have that huge jump in recognition rate. This is recognition reported by the subject. In terms of their image identification accuracy, there is also a big jump driven by disambiguation. The first thing we wanted to know is where in the brain can we find neural activity patterns that distinguish pre-disambiguation MONI pictures versus post-disambiguation MONI pictures. And to do that, we use the searchlight decoding approach, moving a sphere across the brain to ask where in the brain can we use local activity pattern to predict whether the image is shown pre or post-disambiguation. We found that we could do that in widespread brain regions. But this perceptual state decoding might include things that are not specific to the specific image content. It might also include salience or attention driven by having recognized that image. So we wanted to see how neural activity represents specific image. To do that, we use representation similarity analysis, where we take every image and line them up from number 1 to number 33 in the pre-disambiguation stage, in the post-disambiguation stage, in the grayscale stage. And each element here is a neural distance in a particular brain region between those two images. To do the neural, to get at the neural distance, we simply look at the 1 minus Pearson's R of the activity pattern recorded under the two images. This is what we see in V1. We find that in the post-disambiguation stage, the across-image distance in the neural activity is much enhanced. And also there are these blue diagonals in the pre-post pair and in the post-gray pair. So what that means is that this is the same image presented pre and post-disambiguation, and this is the same image presented in the post-disambiguation, and it's matching grayscale image. The distance between images increases in the post-disambiguation stage significantly, as shown here, by averaging across all these elements and compare the mean between pre and post-disambiguation stage. If we look at these diagonals, we find that post-disambiguation images are represented more similarly to the grayscale picture than the same image presented in the pre-disambiguation period. And interestingly, even in V1, we find that the post-disambiguation picture is more similar to its matching grayscale picture than the same picture presented pre-disambiguation, suggesting that prior knowledge and perceptual outcome outweighs the influence of stimulus bottom-up input, even in V1. The big shock came when we looked into frontal parietal and default mode regions. In a nutshell, we found the same pattern, but even much more dramatic than in visual regions. So we find that the between image distance substantially increases in the post-disambiguation period. And for each individual image, they become much more similar to the corresponding grayscale picture than the pre-disambiguation MONI picture. So for example, this is what we find in posterior cingulate. Every dot represents one specific picture. In the pre-disambiguation period in blue, in the post-disambiguation period in green and yellow, and the grayscale picture in red. What you can see is that even in posterior cingulate, neural activity patterns represent post-disambiguation Moni images more distinctly than their counterparts in the pre-disambiguation periods. When we compare the representation format across all these regions, we find an interesting hierarchy. So this shows us in the representation of this set of images how different brain regions relate to each other. And we find the hierarchy from V1, V2, V3, V4, higher level visual areas, and then frontal parietal regions and default mode regions in a way that mirrors what has been described using resting state activity with frontal parietal regions sitting in between default network and sensory areas. So lastly, in terms of the neurodynamic in this task, it's a very interesting question to probe because the reaction time in the task is very slow. Even in the post-disambiguation period, recognized images take about 800 milliseconds for the subject to respond. That suggests that large-scale recurrent neurodynamics is likely involved in the task. We recorded MEG activity in the same paradigm, and this is behavior result. It's very similar to the fMRI data set I showed you earlier. When we look at perceptual state decoding, we find that we can decode perceptual state about 300 milliseconds after stimulus input using the SCP band. And in the classic ERF range, less than 35 Hertz, we have a similar pattern, but the decoding accuracy is lower. There's no pre-stimulus decoding here because pre- and post-disambiguation designation is a stimulus condition that is completely randomized in the task. So the brain has no way of knowing before stimulus presentation which one is going to be. Let's come back to the representation dissimilarity matrix and look at this diagonal in the between condition comparisons. Here we can look at the same image presented pre and post disambiguation, or a Moni image with its matching grayscale picture, and look at neural distance between them. And we find that this is akin to single trial decoding. And we find that between pre-disambiguation and grayscale picture, the single trial decoding goes up after stimulus input and stays high throughout stimulus epoch. Between pre-disambiguation and post-disambiguation Moni picture is the same identical picture, but their distance, the neural representation of them start diverging after stimulus input and reaches peak at around 500 milliseconds. And between grayscale picture and post-disambiguation picture, that is a really interesting comparison shown in blue here. And it shows that they are distinct pictures. And within the first 500 milliseconds, neural activity is very different between them. But after the first 500 milliseconds, brain activity treats them identically. So this shows us the temporal evolution of prior knowledge guiding conscious perceptual outcome. Lastly, because we have both MEG and fMRI data using the same task, we can bring them together to understand how does each information processing stage evolve across space and time in the brain. To that end, we constructed different model RDMs that suggest what the RDM should look like if a neural process only cares about stimulus processing, or only cares about subject recognition, or only cares about attentional salience. So if we look at how MEG activity represent these different models, we find that stimulus activity goes up quickly after stimulus input, showing orange here, and then there's a second bump that might correspond to recurrent feedback activity. Whereas recognition onsets around 500 milliseconds and stays high, and attention appears to have a transient effect here as a switching between stimulus and recognition processing. Now, by taking fMRI data into the same framework, we can ask, how does each information processing stage evolve in the brain across space and time? And we find that for stimulus-related processing, there is a feed-forward sweep with early visual regions activated first and higher order regions activated later, whereas recognition-related activity onsets first in high-level visual areas and frontal-parietal regions, and then move down to early visual cortex, and attention-related activity localized in frontal-parietal regions. So to summarize, we found that prior knowledge impacts perceptual processing throughout the visual hierarchy, from early visual cortex to frontal-parietal areas. And the temporal dynamics of this process is consistent with slow recurrent neural activity across large-scale networks. Lastly, by combining multiple modalities in the same analysis, we find that recognition-related neural activity onsets in higher order regions and then spreads into early visual cortex. To summarize, I showed you 3 different studies related to conscious perception that all point to the fact that conscious perception, the idea that conscious perception results from an interaction between sensory input and the pre-existing brain state. We found that neurodynamics during conscious vision can be described as initial state-dependent, robust transients. They are distinct, spontaneous processes that influence conscious perception in doubly dissociable manners. And past experience can influence your conscious perception in a way that recruits large-scale recurrent activity. So I'd like to leave you with this slide to conclude with a message that sensory input interacts with the pre-existing brain state to drive behavior and awareness. And an important question, I think, for future research is, how does perception and cognition emerge from a brain that is constantly active? I'd like to finish by thanking all the people who did the work. Alex and Brian did the trajectory processing study I showed you. Ayla Potvani spearheaded the separate spontaneous processes influencing behavior study. Carlos, Matt, and Richard carried out the Moni image studies. Thank you for your attention.